Okay, so here we are with chapter 24. As you look at the title, it says the origin of species. So now we're going to start talking about how speciation occurs and the situations in which it does. So what do we mean by speciation? Just real quickly here, it means that you have, so if we talk about species X diverges, which means if they get separated in some way, they can become species Y and species Z. So we would say that species X was the common ancestor of that. So we're going to talk about the things that could make that happen. All right. And so, first of all, species is a Latin word meaning kind or appearance. And so we typically think of a species as organisms that can reproduce and they kind of have traits that make them look alike. So they have some traits in common and they will and are able to reproduce and produce viable or living offspring, and therefore those can reproduce and so on. So this biological species concept. A species is a group of populations whose members have the potential to interbreed in nature and produce viable, fertile, so living, able to have kids, offspring, but do not produce viable, fertile offspring with members of other such groups. All right, and so therefore it separates them out as different species because of that. So in order for speciation to occur, where you get organisms to become two different species, reproductive isolation has to occur. So what is this? Reproductive isolation is the existence of biological factors or barriers that impede, the key here is impede two species from producing viable, fertile hybrids. So a hybrid is the cross between two organisms. So if you have two different species and they try to mate, sometimes they're able to mate, but the offspring are not viable, they may not live, may not be fertile, and so on. So therefore that keeps them as two separate species because they can't merge into one because the offspring just are not that healthy or have some things wrong with it or do not survive or can't have kids themselves, and that's for sure isn't going to keep them from reproducing and so on. And so there are two types of barriers that keep two species separate. So they're called prezygotic and postzygotic. So pre means before, zygotic, the zygote. So this is things that happen before the zygote is even formed. So remember, the zygote is the one-celled baby. Postzygotic is after the zygote is formed. So sometimes the zygote can be formed, but then it doesn't either, it doesn't live very long or grows and isn't fertile, and so on. So our prezygotic barriers, what they do is they impede mating or hinder fertilization if mating does occur. So they're either going to stop mating from happening, or after fertilization, it's going to after mating, it's going to hinder fertilization. And these are all examples of prezygotic barriers, which are habitat, temporal, behavioral, mechanical, and gametic isolation. And I'm going to talk about this habitat isolation first. We're going to look at each one of these separately, but I want to draw your attention to the next page in the top has a picture that shows you an illustration of how everything works. So these are the 5 things that we just looked at. This is the prezygotic barriers. Impedes mating or hinders fertilization if mating does occur. And so here are these 5 that we're going to talk about here. They're all prezygotic. So here's fertilization. Fertilization is where the zygote is formed, right? So all of these things that occur before the zygote are formed is this prezygotic barrier, all right? And so notice here that it doesn't mean that mating can't happen. So here's the mating attempt. So there are some things that stop mating from happening in the first place. That would be prezygotic, before the zygote is born. But then after mating occurs, there's some other things that prevent the zygote from forming. So we're going to look at these. Then we'll look at after fertilization. There's some post-zygotic barriers that occur. So let's say the zygote does get formed. There are some things that prevent it from developing into a healthy, viable, fertile adult that can reproduce and so on and so forth. All right, so let's look at these pre-zygotic barriers. The first three we're going to look at, the first one is habitat isolation. These three that we're going to look at is before the mating attempt. So what prevents them from mating? So the first thing is habitat isolation. Two species encounter each other rarely or not at all because they occupy different habitats, even though not isolated by physical barriers. So they don't have to have literally a physical barrier, but because they live in two different areas, then they rarely come into contact with each other, and so therefore are not going to mate. So you can use this example before. You can have a certain species of deer here in a forest in the Lower Peninsula here in Clarkston, and you can have another population of that same species of deer, but in another place, like in the UP. These guys are not going to mate, all right, because they just are not going to come into contact with each other. It'd be very rare. So another example is two species of garter snakes in the same geographic area, but one lives in the water and one is primarily terrestrial. So this is what it means by even though not isolated by physical barriers, these two snakes really could come into contact with each other, but because of where they live, they're still in close proximity to one another, but because where they spend most of their time, they just don't run into each other that often, and so therefore do not mate, and so it kind of keeps them separate that way. So that's habitat isolation. So that's obviously keeping a zygote from being born because they're not going to come into contact with each other and mate. Underneath the picture on the back that we just looked at is our second prezygotic barrier, which is called temporal isolation. The species that breed at different times of the day, or sometimes breed in different seasons. They have different mating seasons, or sometimes different years even, cannot mix their gametes. So if they're not breeding at the same time, they can't, their sperm is not going to fertilize the egg, and you're not going to get a zygote formed, hence the prezygotic barrier. So temporal, think of it, they're breeding at different times. So think about the tempo, it's the timing of in music and so on. And so that's why they call it that, they're breeding at different times. So example, in North America, the geographic ranges of the western spotted skunk and the eastern spotted skunk overlap. So they don't have habitat isolation here. So they're living in the same range. So they do come into contact with each other, but they don't mate. And why? Because the western mates in late summer and the eastern mates in late winter. So they're mating at different times, and so the hormones and stuff that are elevated at the certain times of the year to make them want to mate are at different times, and so they don't want, when this one's ready to mate, this one doesn't want to, and so therefore this one mates with the other ones, the other western skunks, and these ones mate with the other eastern skunks in late winter. And so that's an example of timing. Another example of temporal isolation could be flowers. So like certain flowers might open in the middle of summer, and that's when it has the pollen and the egg and so on to be fertilized. And then another one is an early spring flower. And so they might live in the same flower garden, but because they don't flower at the same time, they're not going to mix their genes. The sperm from one is not going to fertilize the egg from another. All right, and so here we are in our picture. Here's temporal isolation. So all of this is keeping mating. Mating hasn't even occurred. So the last one is behavioral isolation. Behavioral isolation is more with animals. So we have courtship rituals and other behaviors unique to a species are effective barriers. So, for instance, a courtship ritual could be doing a certain kind of dance. The blue-footed boobies, which are birds, may only after courtship display, and so they kind of move, rock back and forth on their feet, and so on. I'll show you a video of that on Monday. But they look for certain behaviors in their mate, and if the other organism doesn't do that particular behavior, then they won't mate with them. And so that would be behavioral isolation, where a particular behavior is isolating and therefore keeping individuals from the same species mating and not mating with others. All right, behaviors. All right, and so that's all of that is before the mating attempt. So now let's say, now the next two, mating does occur, but then there are two things that can keep the zygote from being formed. And the first one is mechanical isolation. So mechanical isolation is morphological differences can prevent successful mating. So this means we're talking about, like, these are anatomical shapes, all right? They're physically not able to mate. So example, the shells of two species of snails spiral in different directions, resulting in their genital openings not being aligned. So literally the parts don't fit together, and so therefore mating cannot be completed. And so they're physically not able to mate. An example with plants, that's with an animal. An example with plants is flowers often have distinct appearances that attract different pollinators. So it could be a bee versus a fly, all right? The two species of monkey flower differ greatly in the species and colors of their blossoms. So because they have different colored blossoms, and the cross-pollination does not occur between the two plants because the pollinators are attracted to the flower by the color, and so therefore if the bee comes and fertilizes one color, it'll get pollen on it, and remember, pollen contains the sperm, so it'll take that pollen to another flower of the same color or the same species, and so you don't get cross-pollination, so because they go to one particular color. And so that would be another example of mechanical isolation using pollinators. So that's mechanical. So they mate, and they attempt to mate, but physically are not able to complete it because of the anatomical differences. Gametic isolation: sperm of one species may not be able to fertilize eggs of another species. So why is that? Remember, a sperm, how does it fertilize an egg? Well, the two cells together, they have to have the right proteins on the surface. And so sea urchins, as an example, release their sperm and eggs into the surrounding water where they fuse and form zygotes. But it's difficult for gametes of different species, such as the red and purple urchins, so these guys are different species, the red and the purple, to fuse. So like the sperm from the purple can't fuse and fertilize the egg from the red. Why? Because proteins on the surface of the egg and sperm bind poorly to one another. So you have your egg and you have your sperm, and so remember, you have various proteins on there, and they have to kind of fit together. And so if the sperm does not have the right proteins to adhere and allow it to fertilize it, it won't. And so therefore, even though you may have different types or species of red and purple sea urchins, both releasing sperm and egg into the same water, that assures that the red sperm fertilizes the red egg and the purple sperm fertilizes the purple egg. And that is your prezygotic barrier, so that's the gametic isolation. So now fertilization occurs and we get a zygote formed. So now we're going to look at our postzygotic. So postzygotic, and so this we have the reduced hybrid viability, reduced hybrid fertility, and hybrid breakdown. So postzygotic barriers prevent the hybrid zygote from developing into a viable, fertile adult. So these are our three here. So let's look at, so we have the three bullets for the three zygotic barriers. So reduced hybrid viability. Remember, viability is ability to live. It's successful at life. Genes of the different parent species may interact and impair the hybrid's development. So an example, so you have these salamander subspecies live in the same regions and habitats where they occasionally hybridize. So they can successfully, they breed and form a zygote. But most of the hybrids do not complete development, and those that do are frail. So most do not complete development, so they die before they complete development. So that's not good viability. So, and even if they are able to live, they're very frail and have health issues. And so that's not a good viability. So that's why it says reduced hybrid viability. Hybrid is between the two subspecies. Here, the salamander subspecies. So it's a hybrid between two organisms. It's reduced, though, the viability, because it may be able to live, but it's not great, all right? And so therefore, they usually will not create a new species because the hybrids are just have some issues. All right, so the next one is reduced hybrid fertility. So what does this mean? Reduced hybrid fertility, even if the hybrids are vigorous. So even if they doesn't happen what's happened up here where they are alive, they seem to be healthy though. The thing is that they may be sterile, so they can't have kids. And so then they can't form a new species. This new hybrid can't be a new species because a species has to be able to interbreed and have kids to maintain the species. And so this can't be a new species. And so that's a barrier as well. So example is the hybrid of a male donkey and a female horse is a mule. So I'm sure you've heard of mules. And a mule is robust. It's able to do work. It's, it's okay health-wise, but the thing is it's sterile. And so one mule can't mate with another mule, and so therefore that's not considered a separate species because it's not able to pass on its genes and reproduce itself. On the flip side, a hinny is the offspring of a female donkey and a male horse, and is also sterile. And so hinny and a mule, all right, depending upon if the mom is male or the mom is a donkey or a horse, and vice versa with dad. Okay, and so that's reduced hybrid fertility where they are not able to be fertile, the hybrids. And lastly, we have hybrid breakdown. Some first generation hybrids are fertile. Key here is first generation hybrids are fertile, but when they mate with another species or with either parent species, offspring of the next generation are feeble. So again, some health issues, they're weak, not very strong, and they're sterile. So hybrid breakdown. So you have species X and species Y, they mate, all right, you get your hybrid. Hybrid seems to be fine. All right, so that's what it means by the first generations. Our hybrids are fertile. So these are fine, they're fertile, they're able to mate, and so but when they mate with either another species or if they go back and mate with their parent species, then they start to see some issues in the offspring. So it's only after the second generation. So the first generation seems fine, but as they keep on having kids, there's more and more issues, and so on. So an example here I put is cultivated rice have accumulated different mutant recessive alleles at two loci in the course of their divergence from a common ancestor. All right, so here we go. Common ancestor. They diverge from that. And hybrids between them are vigorous and fertile. So you have these two species. And when you cross them, their hybrids are vigorous and fertile, like I was just talking about here. The hybrids are fine, but plants in the next generation carry too many of the recessive alleles, and they're small and sterile, and therefore we have a breakdown in health as these hybrids breed. And so therefore these don't really become new species ever because they're just not successful, they're not healthy, and are not able to have children themselves. And so that is the three things that occur post-zygotic to prevent viable fertile offspring. All right, and so organisms of the same species usually don't have any of these and are able to produce viable fertile offspring. So this keeps organisms a separate species. The biological species concept, though, there's some exceptions, does not apply to asexual organisms. So if they produce asexually where you don't get a new combination of genes, then this doesn't really affect them. Doesn't apply to fossils or organisms where little is known regarding their reproduction. Okay. So I want to do a little bit of the second section here about the process of speciation. So this one was about reproductive isolation, of what keeps species isolated, the pre- and post-zygotic barriers. And now let's talk about how do they actually become different species then. So notice this here, it says speciation can take place with or without geographic separation. So separation can occur in two ways. There's what we call allopatric speciation and sympatric speciation. And it basically is whether they're in the same area or not. So here we have a picture, and this is not color-coded, but I'm going to color code here. So here's one lake, and all these fish, I'm going to color code them all orange. So that's representing a species. And so now, these are all the same species. And we're talking about speciation. So this species is going to diverge and become two different species. So how can they do that? So the first one is allopatric speciation. If you can see here, what happened to the lake? It dried up and there's a land bridge between them. So now these guys can't mate with these guys. And what happens is then they can't mate, they can't interchange their genes. This one adapts to this environment, this one adapts to this environment, and eventually they accumulate enough differences that if you put them together again, they will not mate. And so therefore we would say that they have diverged into two different species. So here's, let's say, the original population in orange, and over here I'll use a different color, I'll use blue. These fish are now a different species, and they will not interbreed anymore. This one, notice here, they're in the same area, so therefore they still are around. So in this case, there's not a geographic separation, but you can still diverge. And this is called sympatric speciation. And so we're going to have these guys here, the blue. So they diverge into different organisms, so with or without a geographic separation. So this allopatric speciation, we're going to look at first. All right, and so how does this happen? So in allopatric speciation, gene flow is interrupted or reduced when a population is divided into geographically isolated subpopulations. And that's what we see here. So one or both populations may undergo evolutionary change during the period of separation. So as I said, they can adapt to different environments, and so therefore what the frequency of genes can differ and start to diverge and so on. So a real life example of this is with these these squirrels. So we have two different species of squirrels, and so here's their names here. And this is a picture of the Grand Canyon. So this is the Grand Canyon, and these guys are physically separated now on either side of the Grand Canyon. So this one right here this species inhabits the south rim of the Grand Canyon. And this one here inhabits the north rim. And so they physically are separated from one another. They're very closely related, though, by testing their DNA. So at one time it's thought that they were one species and got separated so they're physically separated, kind of like here, physically separated on either side of the Grand Canyon, impeding them from being able to reproduce. And so therefore, since they were separated, not able to reproduce, then they diverged into two different species, so that now they in these two different areas, they accumulated by adapting to their environment and what traits were beneficial and so on. They've accumulated enough differences that if you put them together now, they actually don't mate. They have some of those isolations and those barriers that we talked about where they don't mate, which then keeps them even further apart. And so in this case here, what I'm saying here is that they started out as species X, and let's say this is species Y and Z. We'll just call them this is their real species name, but to give you an idea. So then they are separated and diverged into these two species. Why? Because they were physically separated. So the species diverged into two different species. Interesting enough, across the Grand Canyon, they found that this didn't happen in studying species of birds that are found on both sides. Why? Because birds, there's not a barrier, because birds can fly across the Grand Canyon and they still mate with one another, and so they never had that divergence into two different species. So that they have to have that reproductive barrier. So to determine if allopatric speciation has occurred, you have to have reproduction isolation. Reproductive isolation must have been established, and that clearly is the case across the Grand Canyon. So, can we reproduce this in the lab? And that's what this is all about. All right, so I'm going to write on top of here. Let me fix my. So this is figure 24.7 in your book, and it's an inquiry. It's based upon what's going to happen. So the inquiry question is, can divergence of an allopatric, I should say of an allopatric fruit fly populations lead to reproductive isolation? This is an interesting study. So let's look at what they had. So here's the little flies, and this is food for the flies. All right, so this is the initial population. Initial fruit fly population. And what the experimenters did is they took some of this population and put them in a couple jars, and they gave them different food sources. So they took some of them, and some were raised. Some flies were raised on starch medium, which means that starch was their food. And then they took some of the original population and raised them on maltose. On maltose medium. So all they did is, from the original population, is change their food source. And then they did some mating experiments after several generations. So they let them reproduce after several generations and then did some mating experiments to see, did changing the environment, in this case, the environment was changed just by what they're giving them to eat. So that's their environment, what's available in their environment. Changing the environment affect the way that they want to mate. Did it lead to reproductive isolation? So did these flies here want to mate with the Maltose flies if you put them together? Or do they want to mate just with their own cells? All right, so these are mating experiments after several generations. Okay, so, and these are the results. All right, so let's, this is the experimental group, so let's label this mating frequencies in experimental groups. You can write out experimental, or group, I should say. And this is the mating frequencies in your control group. Okay, so what did they do? So let's say, so these were the female flies. And females, there were some females that were raised on starch and some that were raised on maltose. So females from starch, females from the maltose population. And then over here is going to be the males. And some of those males were raised on starch and some were raised on maltose. So maltose and starch. So what they did is they put these guys together to see how often mating occurred. So in this case here, they put a female raised on the starch with the male raised on the starch. So male and female from starch. And they got 22 matings. Then, so it's the same medium. And let's do, so what they did is they took the male from the starch and the male from the maltose, or female from the maltose. So they took one of each, so different mediums, and they got 9 matings. Then they did the same thing, but switched them. So the maltose, male maltose, the male from the maltose, female from the starch, mated them, and that occurred 9 times. And then maltose with maltose, so 2 male and female both from the same medium, same food source, and they got 20 matings. So what they discovered here is, oh, well, look at here, that that they seem to choose and want to mate more often with members that are feeding off the same, that their environment is the same, they have the same food source. But to make sure that it was really the food source, and not whether or not they were in the same population or not, they did this control group. So what does that mean? Low starch with starch. So how do we know, like if they're in the same jar together in the same population, that it has to do with just being in the same population? It really doesn't have anything to do with the food source. Or does it matter, like these guys are in different populations, all right, starch and maltose, does it matter if it's in different populations? So maybe that's what it is instead of just the food source. So in comes the control group. So here's the control group, the female, and instead of doing different mediums, they just focused on whether or not they're in the same population. So females in the same population or different population, and males, different population and same population. So they took a female and a male from the same population. So and all of this was on one medium. They didn't have starch and maltose. So let's pick one. Let's say it's maltose. So same population. So male and female from this jar, let's say. All right. And what's they found? They counted 18 matings. All right. Then same and different. And they got 15. Different and same, they get 12 matings, and all from different populations, 15. And so they took from different populations all the same medium, and what they found is that it seemed to be pretty equal. It didn't really matter if the flies were from the same population or from different populations, that the mating seemed to be close to being equal here compared to over here. So their conclusion here was that when they separated these flies, this represents a physical separation. So that's like allopatric speciation, where they're physically separated. They come from the same population, physically separated. These guys, if they're unable to mate, all right, that's like these guys here. So this starch and this maltose, starch and maltose, are not able to mate. They're physically unable to mate. They end up mating less often when you put them together. Same thing with when we switch up male and female maltose and starch. And so what we started to see is we start to see a divergence, that they're not wanting to mate. So we start seeing, so the conclusion here is a reproductive barrier is forming between the populations of flies on different mediums. It's not a complete reproductive barrier, because you can see on the different medians they do still mate. There's 9 and 9 here, so there is some mating here. But remember, this is over a short period of time. This is just after several generations, which for a fly, the whole fruit fly's life cycle is 2 weeks. So we're talking several generations is a couple of months. And even so, after reproductive isolation for just a couple months, isolation physically, after just a couple months, we start to see this reproductive barrier where they're starting to not want to mate with one another. And so if this continues, it may get to the point where they have diverged, these flies in these different populations, so much that then when you put them together, they might not mate at all, which then can lead to speciation. And so that is called allopatric speciation. So let's look at sympatric speciation. So let's go back to this other picture here. Sympatric speciation is speciation that occurs when the organisms don't have to be separated from one another. So there's not that reproductive isolation that we saw here where they were physically separated. So sympatric means same country, so they're in the same area. So in sympatric speciation, speciation takes place in geographically overlapping populations. So they're not separated physically. Polyploidy is the presence of extra sets of chromosomes due to accidents in cell division. So like non-disjunction, where the chromosomes don't separate properly, would be an example of that. And so remember I talked about before that plants can live with extra sets of chromosomes. So they can be 3N or 4N, and they don't die, their cells don't die like our cells do. So this has caused the evolution of some plant species. So I'm going to elaborate on this that I talked about earlier with plants. So let's turn it over here. And so what we have here, an autopolyploid. Auto means self, so this means that you can become polyploid with just one organism. It doesn't need two. As an individual with more than 2 chromosome sets derived from one species. And so let's look at this, and I'm going to do some color coding here. So there's a chromosome pair, all right? So that's 1 pair of chromosomes. This is 2 pairs of chromosomes and 3 pairs. So this is 2n equals 6, so a total of 6 chromosomes, 2 of every kind of chromosome, so we have 3 pairs. So notice here a failure of cell division in a cell of a growing diploid plant after chromosome duplication gives rise to a tetraploid. Tetra is 4. So 4 of every kind of chromosome, branch or other tissue. So what in the world does that mean? All right, so you have this random cell. So let's say you have a plant. Here's your plant. All right, here's the stem. Here's a branch coming off of that. So notice here it says failure in a cell of a growing diploid plant. So it's diploid. All the cells are like this. After chromosome duplication occurs. So what happens here is the chromosomes duplicate, but the cell does not divide. Oops, now you have double the chromosome numbers. That's how we get a tetraploid. So that means that these guys replicated, didn't separate properly, and now they're all in one cell. All the chromosomes went to one cell. Same thing with these guys. All right, so all the green ones, and the same thing with these guys, all the orange ones. And so now we have a tetraploid, 4n equals 12. Now we have 12 chromosomes, and there's 4 of every kind of chromosome. And this can occur on a branch. So let's say this is the cell that's going to divide, so I'm going to make this one, obviously the cell in this branch is going to be much smaller. But let's say the cell is supposed to divide and make more 2n cells, but the mistake occurs in that cell. So then when the cell we end up with a 4n cell. And so then what happens is this 4n cell is going to continue to divide and make more 4n cells and more 4n cells, and pretty soon we have a whole branch where all the cells are 4n. All right, so then you have this branch coming out of there, and let's say off the branch you get a flower. So remember the flower, so we have a little flower here. So what happens is, let's look at the flower here. Remember the flower, here's the flower. It has an ovary where there's the egg. So inside of here, I should have made that a little bit bigger. Inside of here is a 4n egg. And then coming out around the flower is your pollen. And you're going to have, let's do here, you have here 4n, it's going to be, sorry, it's supposed to be 2n pollen or sperm, because when that tetraploid branch goes through meiosis to make the egg and sperm cells, let's change this to 2n, it's going to be 2n. And that's what this is showing you here. So you have this 4n branch totally from a mistake in a cell. The chromosomes don't replicate, or the chromosomes replicate, but the cell doesn't divide. And it divides and divides and divides, and that mistaken cell, and you get this whole branch that's 4n. Then it produces a flower that's 4n, but that flower is going to go through meiosis. So this is what this is showing you here. So this is meiosis. Gametes are produced, so this is the gametes, the egg and the sperm that I just drew here, are produced by flowers on this tetraploid branch, this 4n branch, and they're diploid because they reduce the chromosome number. So this happened correctly this time. So now when we reduce the chromosome, you have 4 of every kind of chromosome. When we reduce it by half, now you have 2 of every kind of chromosome. So that's what it did. Okay, so now we have these 2n sperm and 2n egg. So then if the sperm goes and fertilizes, the 2n sperm goes to fertilize the 2n egg, guess what? We end up with a 4n cell. So therefore, this gamete, this is what both the egg and the sperm look like this. So this is the egg or the sperm here, that's 2n. And so when so plants, remember, can self-pollinate. So that's what I just drew here. So they self-pollinate, and the 2N sperm fertilizes the 2N egg, and you end up with 4 of the orange chromosomes, 4 of the green chromosomes, and 4 of the blue chromosomes. And this then can start to divide and produce a totally new species. So when this sperm fertilizes this egg, it's going to form a little zygote, and then this is going to form a seed. And the seed then can go under the ground. So the seed is 4N, and then it can begin dividing, and you can end up growing from that seed a 4N plant. And so that's what's going to happen to this here. So you're going to get a tetraploid plant. And this is a new species. And so new species. Oops, I'm off the thing. I'm sorry. Tetraploid plant, new species. And so we could say here that this is called autopolyploid because what really happens here is one species gives rise to a new species. One species does. That's different than if we have 2 species cross and make a new species, which is what this second example is. An allopolyploid is a species with multiple sets of chromosomes derived from different species. So in this case here, we're going to have 2, I'm going to compare this up here, 2 species give rise to a new species. So we start out with 2 species. So that's what we're looking at here, species A and species B. All right, so here is species A and their chromosomes. I'm just going to keep species A the same color to distinguish between what came from one species and another. So species A has only 4 chromosomes in all of its cells, so 2N equals 4. So it's diploid, but it only has 4. Species B, on the other hand, has 3 pairs of chromosomes. So I'm going to color them all green. And so they have a total of 6 chromosomes, so 2N equals 6. So notice here, we have a mitotic error. So this is mistake 1. There's 2 mistakes that are going to occur in here. So mistake 1, where the mistake occurs is in this species making its gamete. Notice here it says unreduced gamete. So the chromosomes replicate, but the chromosome number is not reduced to 2N. So again, non-disjunction occurs and the chromosomes don't separate properly. You're supposed to only have 1 large chromosome and 1 small chromosome in here to have, instead of 2N, having just N. So a mistake happened, and now we have their gametes having 4 chromosomes. So the gamete is 2N. It's supposed to be N. Oops. Species B does things how it's supposed to do it. So species B reduces its chromosome number. This is meiosis right here for species B. It reduces it. The normal gamete is 3, so it has 1 of each kind of chromosome, and so that happens properly. So now what happens is, let's say this gamete, let's say this is the sperm, fertilizes the egg. So now we have a hybrid between these 2 species. Oops, so let me get, so these guys right here are from, these are the 2 from species A, and these are the 3 from species B. And so if we notice here, these guys aren't matched. They're hybrids with 7 chromosomes, so they don't have 2 of every kind of chromosome because these guys don't have a match, so it's kind of a weird thing going on. But the plant can grow, the cell can divide and grow into a plant, and that plant can produce flowers and then start to produce their own gametes. So now notice here, this now is mistake number 2. So now you have this weird hybrid. And mistake number 2 is when this hybrid goes to make its gametes. It's, again, another mistake on reduced gametes, so it didn't reduce the chromosome number by half. It still has the same chromosome number. So it has these 2 here, and it has these 3 green. So it did not reduce it. So it still has 7 chromosomes. Another mistake. So now this is a gamete. Let's say this is an egg. So let's say now so now you have this weird species here, or this organism here. So this is an adult plant right here, and it produces this egg that didn't get reduced. And so all of these guys are living, let's say, in the same meadow. So you have species A, some flowers like that, species B, some flowers like that. Now you have this hybrid that's in the middle between them. And so what happens is, it's possible that species B, when they make its gamete here, makes its normal gamete, reduced to 3, n equals 3, there are 3 chromosomes, that this sperm from this plant, the parent species, can fertilize this egg from this hybrid here with this abnormal number of chromosomes. And what happens then is, look at these 3, when it fertilizes this, match up with these 3, and then all of a sudden you have pairs of chromosomes. And so these guys here are their original orange. And now these 3 match up with these 3, and now you have pairs of chromosomes. And so now you have a hybrid that is diploid, 2n equals 10, 5 different types of chromosomes, 2 of each kind, and this can grow and divide, and all of a sudden you have a viable fertile offspring, and this, we would say, is a new species. Different than A or B. All right, so this is a totally new species. This one in the middle is not a new species because the chromosomes don't have pairs, and so they can't really, like it. This is a mistake. They don't reduce their gametes, but if they try to do meiosis, they don't have pairs of chromosomes here, and so they can't, this is not considered a new species because then the gametes would have odd numbers of chromosomes and so on. All right, so this is not a new species, but this, if this happens, is. And so those 2 are examples of either happening in one plant or 2 plants living right in the same area, giving rise to a whole new species, and they don't have to be separated, which by definition is the example, or is sympatric speciation. All right, so sympatric speciation, oftentimes we see it a lot with plants. All right, and so with these polyploidy and so on, giving rise to new plants. All right, so this is where I want to stop. All right, and so we'll go over the rest of this probably on Tuesday because we're going to do our activity on Monday. Have a great weekend.